And I believe Alex is ready to go on with his talk. So apologies for anyone who needed a comfort break. You need to hang in there for a little bit longer. So while Alex gets his screen up, Alex had his PhD from Florida State in 2016, working with Scott Stagg, software development for cryo-electron tomography, as well as contributing to various biological projects. He's now a postdoctoral fellow at the Simons Electron Microscopy Center, part of the New York Structural Biology Center, and he's working on cryo-FIB-SEM development and deep learning applications. I think we're about to hear about some of these deep learning applications. So thanks, Alex, whenever you're ready.
Okay, great. Can you hear me and see slides and everything?
I can see your slides, I can hear you.
Wonderful. Okay. So thank you, everybody. Yes?
太多了，东西内容太多了，我们做的东西太多了。
Can you please?
稍微垮了点。
Sorry, can other people mute? We're getting noises off.
Okay, great. So I'm going to try to cure people of reply or asking questions in the chat by having a bit of interactivity here. All right, so I'm going to be talking about neural network particle picking and denoising with Topaz. So Topaz is a piece of software written primarily by Tristan Bepler at MIT in the deep learning lab of Bonnie Bepler. And you can find publications in Nature Methods and more recently in bioRxiv for the denoising, and software is fully available. I'll just tell you up front. So why in the world are we having a particle picking talk? Haven't we solved this long time ago? Well, let me ask you all, do you have problems with your micrographs having, let's say, aggregation, or you have weird shaped particles, or you have small particles, you can't even really identify them in order to pick them completely? So the interactivity part here is if you have any one of these problems with any of your current samples, please type some combination of 1, 2, or 3 in chat. I like to have a good idea of the types of problems people are having and who I'm talking to, and keep your questions in the Q&A. So this will flood out the questions inside of the chat. All right, so while you go ahead and type that, I'm going to give you a quick overview of what I'm going to talk about today. I'm going to talk about how Topaz picking is beneficially different, what types of problems Topaz solves, and I just gave you some hints there. I'm going to go over some examples, go over neural network denoising and how it can actually be useful for you, and then quickly talk about our current work. So I want to open up my chat. I see got 100 replies here. Quickly scroll through, 1, 1, 2, 3. All right, lots of 1s, lots of aggregation problems, good amount of 3s, small particles. Okay, so hopefully this talk is actually useful for you because all this software is available and actually integrated. I'll show you here later. All right, so current particle picking methods. Conventionally, we've always used some form of template picking or rotational averaging or difference of Gaussians or Laplacians in order to try to geometrically, at least in 2D, identify particle locations. More recently, we've had some positive negative convolutional neural network softwares come out, including Cryolo, Warp, Deep Picker, and these either implicitly or explicitly require that you label what things are a particle and what things are not a particle in training the neural networks. Now, Topaz here is different. It uses a new neural network framework that is positive unlabeled. So as you may note by there's no negative here, you don't actually have to know where your particle is not located. So it removes some of the practical issues of actually training the model on your particular data set. So conventionally or conceptually, how does this actually look? So we have a bunch of particles, and they might be, we're going to call them pluses here. And then we have a bunch of non-particles, we're going to call them negatives here. And as you see in this micrograph, we have all the conventional issues that I talked about before, the 1, 2, 3. We have aggregation, we have this weird shaped particle here, and we have some particle views that are quite small. Can we solve this problem with Topaz? And this is how Topaz solves it with positive unlabeled picking and training. So all you need to do pretty much, is identify several of your actual views that you could see in your micrograph. So you don't have to identify them all. You can incompletely pick and it works fine. And you don't have to identify any negatives. So it actually solves the hard problem of not knowing where all your particles are a priori in your micrographs. And then Topaz is specially designed to then take those positives and differentiate them from the other positives that you have not labeled in the micrographs that you're training on, and also differentiate them from junk and aggregate that are actually real or true negatives. Okay, great. So what are some things that Topaz solves? It assumes sparsely labeled training data. Like I just mentioned, you don't even have to label all your positives. You can label just a few, and no negatives. You can pick, in our experience, any size and shape particles. You can pick more real particles than any other pickers that we've tested against on several different data sets. I'll show some here. We have in Topaz a very high ratio of true positives to false positives, and this allows for reduced bias and classification filtering. So you're not throwing away good particles in your junk classes. You're just getting primarily good particle picks. And surprisingly, something we found is that Topaz centers particles quite well, and this actually reduces them falling out of classification. So you could maybe try to exhaustively pick an entire micrograph, but actually Topaz will pick better than that exhaustive picking, because then you're still filtering through the junk and the non-well-centered picks. Okay, great. How well does Topaz actually work? So as I've shown before in previous slides, there's this weird shaped particle that we had. So this was in collaboration with Micah Rapp, a grad student over at Columbia, and Larry Shapiro's lab. And this particular particle, it's shaped kind of like a question mark. It's asymmetric, it's non-globular, it's about 100 kilodaltons, and it aggregates. It's got all these problems. And so what Micah did is he just, without even asking us for help or anything, he went to the GitHub website for Topaz and just followed the generic workflow. And how did he actually do? So he compared it with not just template picking and difference of Gaussian picking, but also deep picker and Cryolo, but those didn't pick even about 10% or 20% as many particles as these pickers. So we're comparing between these three pickers here. And as you can see, Topaz in the end picked, I want to highlight, virtually no junk. It avoided all aggregates. The only junk that it actually picked were in micrographs that had black stripes because of Gatan K2 random black stripe issues. And then, as you can see in the middle right two boxes here, it picked on the order of a factor of 2 to a factor of 10 more difficult views, these side and these top views that are rather small in the projections. He went ahead and reconstructed, and without even doing any filtering of junk, he just directly pretty much fed all of the particle picks for Topaz into CryoSpark homogeneous refinement, and then non-uniform refinement. About 1 million particles went in and got a global resolution, about 3.7 angstroms, and actually much better Euler angle distribution than both template and dog picking, which picked somewhere between 60 and 70% or 75% of the particles that Topaz picked. All right, and if we go look in detail at how Topaz did, we can see that Topaz actually is able to uniquely resolve, just by picking more particles in more views, uniquely resolve the secondary structure of this particular, this is a toll receptor, like cell adhesion type molecule, compared to all other pickers. So these are the next best two pickers. All right, so I told you Topaz can sparsely pick, and how sparsely can you actually pick? You can see this in the Nature Methods paper that came out in October. So in that paper, we did various benchmarks, and this is how sparsely we picked on the micrographs that we fed into training. So here's T20S proteasome, and we picked somewhere between 100, I think we actually went down to 10 particles, but we recommend picking at least 100, maybe up to 1,000 particles, but you can only, you only have to pick this few number of particles. So this is just a random sampling of the micrographs that we use to train the various data sets. So here's ADS ribosome here, various data sets in the paper. And we actually only picked down to 1 particle per micrograph. So you don't actually have to know where almost any of your particles are. And one good thing about Topaz is that it ranks particles quite well. So Topaz outputs a scoring value for every single particle that it predicts. And as you increase that scoring value, you can see this is 2D classes in the rows and the scoring value as you go down here. And as you see, when you have the scoring value quite low, so you're picking a low number of particles, you get no junk. And as you increase it and increase it, you're still getting no junk until at some point you start introducing some kind of junky classes over here. So it ranks particles quite well. With T20S proteasome, we had some black flakes from the ultra-AU foil grids that came in here. And so those were filtered out. If you want to filter by scoring, you can filter them out by scoring. And same with aldolase here. Virtually no junk particles picked, no matter the scoring threshold. All right, so I also said Topaz can pick some weird shaped particles. I showed that question mark particle. Here's another particle called clustered protocoherent. It's about 30 nanometers long and maybe 5 or so nanometers in this dimension and this other dimension here. And as you can see in the projection, it's quite low density. It's this weird shape. It's kind of hollow. And so what the data owner did, Julia Brash, again in Larry Shapiro's lab at Columbia, is the blue circles are her labels for where the particles are. And she did her best to exhaustively pick every single particle that she could find by eye, and then trained Topaz. And as you can see, Topaz pretty much misses all the junk, misses the carbon, and it picks several times more particles than Julia was able to even see. All right, and a couple weeks ago, so I get to show you some COVID picking successes here. We went ahead and we're collecting through NCCAT and in collaboration with the Larry Shapiro lab, several COVID data sets with various ligands and antibodies bound. And here we are taking all our team at NCCAT, at SEMC, and in NRAM. Oops, sorry, I'll go back. And they all threw everything they could at the first data set that came out, or one of the first data sets that came out. So they're throwing various templates at picking this particular particle, blob picking. And in the end, they were able to get about 100,000 or so particles as initial picks. And then they went through, did classification, and got a reconstruction with about 10% of those particles. Now we took those 10% good particles that they actually found, fed them into training into Topaz, and then Topaz outputted about 50,000 initial picks. And as you can see, we have much clearer, better angular distribution and almost no junk in any of these picks. And so this only took maybe about 5 or so hours to train and then extract these particles. And as you can see too, this is in the CryoSpark interface. So here's the final results of the Euler angle distribution. About 10% of the particles from template and blob picking by our entire group, and about, let's see, about 60% of the particles went through and were good particles. So remember, this was trained on particles that were initially picked by template and blob, and those actually had some bad particles in it. So Topaz was trained to actually pick some bad particles, but it still did much better. So we got about 150% more particles. And here's what the final reconstruction was, or at least one of the final reconstructions. We've pushed it a bit farther since. So with those 13,000 particles of template and blob picking, we have a resolution of about 4.5 angstroms, and Topaz picking just trained on those good particles, and then repicking got to about 3.5 angstroms. So we're able to do much better therapeutic studies with this increased number of particles in picking. And that actually reduces the amount of classification we have to do. So it makes everything much easier. Okay, great. So that's particle picking. Let's quickly jump into neural network denoising that's in Topaz now. So Topaz now denoises. It uses the noise-to-noise framework previously published, and we have restructured this to work on where you split frames up and use these frames as training data sets. So we've trained it on dozens of cryo-EM data sets. A general model is out, you can use it, you can train your own model. And what it does is helps you directly visualize difficult proteins with confidence, and I'll show you some other useful functions. So how does noise to noise actually work? Let's quickly go over this. So I mentioned what you do is you take your frames and your individual movies of every single micrograph, you split them in half, and then you get two exposures of the same field of view. But now what you have in common between these two exposures is effectively the signal, and what is not in common is the noise, if we're assuming that it's actually truly independent, which it's not completely, but we're going to make that assumption. So you have one noisy image and another noisy image. You train the network, it's a UNet, to learn how to transform noisy image 1 into noisy image 2. And then you do that for many, many, many micrographs, many thousands of micrographs over a different set of data, different microscope, different camera, different collection parameters. So it kind of learns the entire image formation process of your microscope systems that you're using. And so what you effectively get is, when you hand this neural network, now after you've trained it, a new unseen noisy image, now it knows how to transform that into a bunch of different noisy images, but that's not very useful. So what you actually do is you train it to learn how to transform your new noisy image into something like the average or the median or the mode of all those noisy images, which is actually what the clean target is. This is the non-noisy image. And so does this actually work? So here we have a particle in a nanodisk, protein in a nanodisk. And as you can see, the original image looks like this. After denoising, it looks like this. So just visually, it's quite starkly better. Here's proteasome, same micrographs I showed before, and here's the denoised image on the right. And if we go ahead and try to optimally low pass that proteasome micrograph, we get the image on the left, and you can then make a visual comparison of the image on the right in SNR comparisons too. And you can see that the areas where there's no particles are quite flat over in the clean target. So we effectively removed a sheet of noise off these micrographs. Here's some other examples. Here's a ribosome data set, and you can see this high frequency speckles is more or less removed over on the right, and you can actually start making out structure of these ribosomes on an individual basis. Okay, so is it actually useful? It's a nice anti-squinting device, and it actually allows you to look at particles much more clearly. How useful is it? So here's that clustered protocoherent data set as a single particle data set. On the left here are subtomogram averages of the 3D structure. And as you can see in this orientation, when you rotate this 90 degrees, you get just 2 dots effectively in the projection. And so you might go through your micrographs and think, well, is this a top view? I see a bunch of dots in various places. These two are about the right distance away from each other. Is that a top view or not? So if you denoise this micrograph, then you can say quite clearly, this is a top view. Now other top views start jumping out across the micrographs. And what you can do then, which is what I did in the denoising preprint, is I went ahead and retrained after picking on denoise micrographs and trying to get as many top views as I could. And so I trained on about 1,000 particles that I picked, whereas the data owner trained on about 1,500 particles, and went ahead and then trained a Topaz picking model using these particles, and I got about 60% more particles. And as you can see, the Euler angle distribution is much better. Though it's still a difficult structure, it's still low resolution, but I just want to show that denoising micrographs allows you to identify Euler angles much better. Okay, what else can we do with denoising? So it can allow us to be more confident of what is in our image background or our sample background. So what do I mean by that? So this proteasome micrograph I showed before, so if you denoise it, then you have virtually flat backgrounds. We know by tomography that there's actually no protein here. There's no junk at all. So we can use this as a negative control to say, if we denoise and we don't see anything in the background, we can be pretty sure that there's nothing actually there. Whereas on the right, we have a positive control. We have some microtubules with tubulin scattered about in the background, and kinesin, I believe. And then after we denoise it, we still have those features. So we have a negative and positive control. So then what you can do is you can take your new data set, and here in this example, I'm using the Mao et al. HIV trimer data set back in 2013, which is released on Empire. And if you look at the raw images, you might not be sure if you're seeing actually real particles or not. You can go ahead and denoise this, and then you can do closer inspections. And at this point, you can start to decide, is your sample just full of junk, or do you have real particles which might actually exist in this data set? Okay, now last thing, one of the last things I want to touch on real quick is we have now converted the neural network denoising to 3D, and I'm actually now training about 200 gigabytes of tomograms on 8 GPUs. And so this is one of the results afterwards. So here's inside of a yeast lamella we did cryo-ET on. And as you can see, all this speckly information over here is removed. I mean, the tomogram is still filled, of course, with a bunch of stuff because cells are full of stuff. But if you look in between all these proteins and membranes, you can see rather flat, somewhat featureless areas. And so we are effectively denoising in 3D now. We'll have a general model out here quite soon. Okay, so if you want Topaz, you can go to this GitHub website, T. Bepler, that's Tristan Bepler. There's a streamlined install with many different installation methods. There's pre-trained denoising and picking models. I forgot to write picking there. Full tutorials, there's a GUI, and it's integrated into CryoSpark and Appion. And there's going to be about 100 data sets or so released on Empire, about 40 terabytes that we used in part to train and test the denoising functions. So I quickly want to highlight the CryoSpark integration. So they've been working quite closely with me to make sure that this works well, and I've been actually, we've been updating it while I've been processing the COVID data sets. So integrated completely pretty much into CryoSpark, as you can see here, is the denoising functions with pre-trained model. The training functions, you can either use a pre-trained model or you can train your own model. And it's all just simple. You drag over your micrographs, your particle picks, you extract your particles at the end. And as you can see on the bottom right, there's pre-trained models. So the updated version of this CryoSpark release that we've been working on will be out soon also. All right, quick summary. So with Topaz, you can pick more and you can pick more accurately with the positive unlabeled the framework that it uses. With denoising, you can visualize and identify your proteins and your background much more confidently. And you can see the corresponding publication and preprint. All right, I want to thank primarily Tristan Bepler. He's the brains behind this, and his PI, Bonnie Berger. The CryoSpark team, in particular JU, has been very helpful and responsive in integrating Topaz into CryoSpark. Katara Kelly worked on the 3D denoising, and some of the examples I showed came from Micah and Julia from Larry Shapiro's lab. All right, and if you want to hear about those new releases, follow me on Twitter. You'll be the first to know about the tomogram denoising, pre-trained models, and the CryoSpark integration update. Thank you.
Okay, thanks very much, Alex. So again, lots of questions. I'll try to pick a few. So from Michael Sauer, could you explain in more detail how the neural network works differently between Topaz and Cryolo and Warp?
I don't know those fine details. The most I know from a conceptual standpoint is what I mentioned before, that Cryolo, Warp, and Deep Picker, they use positive negative frameworks where they usually either have you pick those negatives explicitly, or they assume that some of the non-picked regions in your micrographs when you're training are negatives, whereas Topaz does not use that assumption. It tries to estimate the fraction of area inside of each of your micrographs when you're training as actual negatives, and therefore it picks up the remaining positives. So I don't have the detail on that. You can look in the supplementary of the picking paper, has a lot of that information. If you have individual questions, you can email Tristan Bepler. He has the knowledge in this area.
Yeah, okay, thanks. So from Magdalena Schachel, what is the size limit for particles that is successfully picked with Topaz?
So there's, yeah. Go ahead.
Sorry.
So there's, as far as I know, there's effectively no size limit, because what you do in the step before training is you downsample your micrographs, and all you need to make sure is that the receptive field of the network that you're using to train is bigger than your actual particles. And if it's not, then you just downsample more. So if you have some giant particles, then go ahead and downsample to 16x the size of your original micrographs, and that should work fine.
I think the question was more about not large particles, but very small particles.
Ah, okay. We haven't run into a size limit yet. So the cluster protocoherent particles, which I highlighted, and I'm going to point to one of the top views here, if you can see my mouse on this screen, is those are about 13 kilodalton individual pieces that make up that that protocoherent. And so what you're seeing there is about a 5 to 10 meter in width and 5 nanometer in the other dimension, kind of a 2 dots. And Topaz can pick those 2 dots just fine. And what we've found is that Topaz trains the best when you make the training radius of where your particles picks are, where you're telling it they are, to be somewhere between 1 and 3 pixels. So you want to only train on very small pieces of your proteins, and that makes it very robust to small sized particles, we believe. We haven't seen a limit.
Okay, thank you. So there were a couple of questions about ice gradients. I've lost who said that, but basically how does the ice gradient affect particle picking and the denoising?
So as you might expect in denoising, when you have thicker ice gradient, then it becomes more opaque even after denoising. I don't believe there's a gradient subtraction in the denoising function. In particle picking, there is an initial step when you're doing downsampling, pre-processing step, that tries to at least subtract out very thick gradients that might be junk or carbon on the edge of your micrographs. And it does so with a Gaussian mixture model. And so it tries to identify 2 humps in a Gaussian and which one is actually the ice and which one's not the ice. And so if you have really thick ice, then that might get thrown away. But other than that, there's no other built-in functionality that I'm familiar with. And we haven't seen a limit in thickness. Usually you don't collect more than 200 nanometers thick if you're really, you have some weird particle that prefers thick ice, and we haven't seen any issues with them.
Okay, thank you. So there are a couple of questions about how, so you mentioned how Topaz integration into CryoSpark. What about getting the particles into RelyOn?
So to get out of CryoSpark to RelyOn, there's a script, I think it's from the CCP-EM people, or maybe it's a different set of scripts, but it's called CSpark to RelyOn, or CSpark to star, something like that. If you do a Google search for CryoSpark to RelyOn conversion, you'll find that script and a bunch of people explaining how to use it.
Okay, and if you run Topaz standalone, is that?
Topaz standalone has a conversion function that'll move stuff from its format to star format or to eman2 format, and there's another format, I forget which.
Yeah. Okay, final question, I think, from Matthew Martinez. Does denoising sacrifice some high spatial frequency info?
Good question, and let me go to the next slide. Thanks for asking that. So on the left here are 3 micrographs randomly chosen and not denoised in the FFT, and on the right is after denoising. And you can see quite an amplification in the lower about 1/3 Nyquist, and then there's flattening across the entire spectrum. And for some reason, we're not completely sure. We think it might have to do with the corners of all the micrographs that it trains on. We get quite a drop off at about 2/3 Nyquist. And so you maybe shouldn't trust any information out there, but really you shouldn't be using denoising to do anything past the initial analysis step of your micrographs and particles and helping to pick. You don't want to feed any denoise micrographs or in the future tomograms into alignment and reconstruction, because there's a general problem in neural networks, which is called the hallucination problem, where you might be introducing false information into your post-processed data because of it being in the training data effectively. And that could be out in these high frequencies. So I would suggest not using those high frequencies, really.
Okay, thank you. That was a very clear distinction. I think at that stage we better move on. So thanks very much.